Welcome to the Encore playback system. Please enter your conference ID number, followed by pound.
Welcome to the Encore replay of the Barclays Speaking the Science call series occurring on April 11, 2019 at 1 p.m. Eastern Time.
Is this the conference you want to hear? Press 1 for yes or 2 for no. Press.
After the tone, please state and spell your first and last name, then press the pound key.
Vikram Khanna.
Recording completed.
After the tone, please state and spell your company name, then press the pound key.
Icarian Capital.
Recording completed.
After the tone, please state and spell your email address, then press the pound key.
Vikram@icariancapital.com.
Recording completed.
After the tone, please enter your telephone number, beginning with the area code, then press the pound key.
Good day, ladies and gentlemen. My name is Ian, and I'll be your conference operator today. At this time, I would like to welcome everyone to the Barclays Speaking the Science conference call series. All lines have been placed on mute to prevent any background noise. After the speaker's remarks, there will be a question and answer session. If you'd like to ask a question during that time, simply press star followed by the number 1 on your telephone keypad. If you'd like to withdraw your question, simply press the pound key. Thank you. I'd now like to turn the call over to our host for today, Ms. Gina Wang. Ma'am, you may begin.
Thank you, Ian, and thank you everyone for dialing in. We are very pleased to continue Barclays 2019 Speaking the Science conference call series. Our call series are intended to focus on key scientific concepts and the progress of a preclinical and a clinical program. Please hold your questions until the end, but as always, feel free to email me at gina.wan@barclays.com if you prefer anonymously. Today on the call with me is Hank Hughes, President of Worldwide Research and Development, and Robert Bassey, Head of Technical Operations from BioMarin. Thank you, Hank and Rob, for joining us today.
Thank you, Gina. We appreciate the opportunity to provide an update. As most of you know, BioMarin is a 21-year-old company, and we have 7 approved products in the market. And we anticipate they'll generate about $1.7 billion in revenue in this year and about $2 billion in revenue in 2020 next year. That guidance includes contributions from Palynziq for the treatment of phenylketonuria, our newest commercial product that was launched in the U.S. last July and is expected to be approved in the EU later this quarter. Palynziq has been well received by the PKU community, and we're very pleased with the pace of uptake in the U.S. since launch. In addition to our commercialized products, we have significant development pipeline, including late-stage products for vosoritide for achondroplasia and Valrox gene therapy for severe hemophilia A. And we also anticipate filing an IND for BMN 307, our gene therapy product for phenylketonuria. So it's with a lot of pleasure, Gina, that we talk to you today, and thank you for the invitation to talk about science and pipeline. With Valrox, we're moving ahead at a very fast pace, having recently checked many of the boxes for a potential accelerated approval, whose decision we've targeted to communicate to you in the back half of this year. A key element of accelerated approval will be efficacy results from a subset of patients in the ongoing phase 3 study, as we communicated at the JP Morgan conference earlier in the year. In addition, the 3-year update from the ongoing phase 2 study will provide ancillary safety and durability data. Given the imminence of these milestones, there's been a lot of interest in the Valrox program, so I want to hand the call back to Gina to address some of your questions. It's also important just to remind you that we have not seen any of the data from the ongoing phase 2 or phase 3 studies, as both studies are being run in a way that the company doesn't see the outcome of the study to avoid the introduction of bias into the study. So as such, please don't attempt to interpret anything that I say as being relevant to the phase 3 ongoing study or the phase 2 3-year data, because I simply haven't seen the data. So with that, Gina, back to you.
Okay, great. That's very helpful. So maybe I would just continue with Varro's question, in hemophilia gene therapy. And I think, this is very exciting. Actually, yesterday we just hosted a call with the payers. Actually, the payer expert sounded also extremely excited about this, and I think in the future could save a lot of the cost for actually payers, the insurance, if, the durability of the gene therapy can really last a meaningful time. So maybe I would just start with, you recently announced the enrollment completion for accelerated filing requirement for Varox. Can you just please remind us what number of patients was required for accelerated filing?
We haven't given an explicit number, Gina, nor explicit description of the statistical measures in the study, and we've done that for largely proprietary reasons. But relatively shortly now, you'll have a better sense of what the game plan is, because as I said, in the back half of the year, we aspire to communicate where we are in regard to filing for accelerated approval. I think one of the most important points to keep in mind about that subset in the accelerated approval application is that the data that will drive their decision is based on factor VIII at a relatively earlier time point after gene dosing. And that's the only statistical testing that's being done as part of the accelerated approval application. The agency has also asked us to provide estimates of bleeding rates based on 6 months of observation of patients at a when they become in a non-hemophilic range. And so all that together says that this is a year that we should be on track for being able to make a decision about filing for an accelerated approval. And the key things to know about are the time points that it can be completed within this year, and that we are representing that we have adequate power to meet the statistical hurdles that the agency requires. Okay.
So you mentioned that, pretty rapidly, after dosing, are we talking about like a 3-4 months, or are we looking more at a 12-month time frame for factor VIII level?
Closer to the first and farther away from the latter. So 23 to 26 weeks is the time point at which the measurement of the factor VIII for the purposes of testing whether we can get whether we can get that meaningful above 40% response.
Okay. So also, I think another thing, 40%, you mentioned that, have you confirmed with the FDA that, factor VIII level above 40% throughout 12 months, or that early phase of the trial, if it hit a statistical significance, would that be approvable just based on the factor VIII level?
Yeah, so we have had that conversation with the FDA extremely explicitly, and the agency has heard our statistical analysis plans and provided input and comment about that as well. So we feel very confident that we're aligned with the agency about how to look at the early accelerated approval cohort for initial registration. I think it bears emphasizing a couple of things. One is their guidance document said in the normal range, and we had some discussion with the agency about, 40 to 50 is a area of some ambiguity. The professional laboratory people would say 49 is abnormal, and the professional hemophilia people would say it's in the non-hemophilic range. So the agency sided with the hemophilia community and said, okay, above 40 IU per deciliter is sufficient. So that's where that 40 came from, and that's the only kind of number we've revealed to the public community about the testing, is that's the target for effectiveness. But we've agreed with FDA on the explicit, p-value and specific statistical tests that employ for comparison. So our agreement, the input the FDA has provided in our agreement with them goes well below in depth just simply 40 IU per deciliter. We called that particular point out simply because there's a little confusion between what's in the guidance and what's in the hemophilia community. But suffice it to say, the agency, basically what they're saying is if we're going to give the opportunity for doctors to prescribe this drug for patients with incomplete clinical effectiveness data. We've got to be convinced. We have to have conviction that the magnitude of the benefit on factor VIII is robust. And for them, that means above 40 IU per deciliter.
Mm-hmm. So, that's the magnitude of the benefit. What about the other side, like a duration of the efficacy? Do they require that, 40 unit per deciliter through 12 months, or would they wanted to see even for longer time? I know part of the accelerated approval package also will include 3-year, data from phase 1-2, and how important that data will be for the accelerated approval decisions.
Yeah, so the agency's view about that, I think, can be summarized as follows. First of all, they've seen our 2-year data, and we haven't changed our plans materially since they've seen our 2-year data, and they understand that there's a 3-year time point coming up, and they understand also that we haven't looked at the data, so we haven't had explicit conversation about how would you interpret your 3-year data. But what I can say is that they obviously know about the 3-year time point, will be informed in their thinking about the 3-year time point, but we haven't had explicit conversation about how they'll be informed in that 3-year time point. What I can further color this a little bit about with the agency, in terms of our discussions with the agency, is they're not very explicit about their requirements for durability demonstration in their guidance document, and partly that has to do with the fact that the issue of durability, it's of heightened awareness in a gene therapy context, because everyone's hope is the pure one and done curative play. But from a regulatory perspective, as a matter of fact, they don't typically require the same level of strength of evidence about durability of drug effect in chronic settings. And so you can sort of see that pattern in the sense that in the accelerated approval context, statistical tests of factor VIII, no statistical test of bleeding. In the confirmatory study, statistical tests of bleeding, but no statistical tests of durability or no requirements beyond a year of follow-up measure of the bleeding rate. So I think what you get a sense of for today is they know what's going on. They see the same things that you see. Their working assumptions are that the durability of Valrox will be assessed in large measure based on the ongoing phase 1-2 study, which is why it's a bullet point. Now, all of our presentations about key to an accelerated approval filing, and that primarily that they're going to be focused on clinical evidence of durability. Their main thrust is not on surrogates, it's on clinical outcome variables. And for them, the clinical outcome variable that's the most significant is the bleed rate. So one way to think about this is that they recognize the urgent unmet need of hemophilia. They're willing and flexible about enabling accelerated approvals based on factor, but their real money shot is bleeding. And I think they get to that place largely through discussions with clinicians. When you ask a clinician what's the number one problem of hemophilia, they'll say uncontrolled bleeding, and the FDA says, great, stop that. So the 3-year data are going to be important to them. Exactly how, remains to be seen, because we haven't seen the data, nor have they. And I believe that the expectation should be more modest in terms of proving durability than a statistical test.
Mm-hmm. So maybe going back to 40% activity, would that be a chromogenic assay, or would that be a one-stage assay?
Well, so that's an interesting question, and we've talked about that a lot, and we've settled on the chromogenic assay, but it's maybe worth recapitulating a little bit of how we got there. Discrepancies amongst the assays have been known for a while, and our particular flavor of discrepancy is actually in a different direction than they're accustomed to seeing. So they just asked a basic scientific question, which is, which of those answers is more reflective of what you're observing clinically? And of course, we can't answer that question definitively, because we need to know about the correlation of transgene factor VIII activity and bleeding consequences. And given how high we can get factor activity, it may be many years in coming before we can recreate the relationship between bleeding rate and transgene factor expression level. So in that context, the discussion around the assay wasn't definitively settle the argument about which assay is the most reliable measure of bleed control, but rather which measure reflects the transgene product that you intended to make. And at R&D Day, we showed last year, we intended to make a B domain deleted, basically equivalent to SQ product, and the assays off the shelf, which characterize our product as a B domain deleted product with the appropriate range of specific activity, is the chromogenic substrate assay. And so the agency liked that also in particular, because if you remember, I said if they're going to do an accelerated approval on a relatively fewer number of patients without clinical outcome statistical tests, they want to have conviction. And so the pivot to the chromogenic enabled them to have a little extra dose of conviction, because if we had come in with the one-stage assay, they might have thought, oh boy, that makes it a little harder for us to have conviction because we don't know what this lower number means. They like the fact that we're using the lower number for accelerated approval. But as to which number and what level corresponds to definitive clinical benefit, or in our case, bleed control, that is going to take years to settle. So what I hope to set up for you was sort of the pure accelerated approval consideration, which has to do with purely accelerated approval, surrogate endpoints, laboratory measures, and the real itch that the agency is trying to scratch at, which is we want to understand how ultimately to interpret an assay against the clinical outcome variable, but we recognize that's years in the making.
Okay. So is it fair to say, like, say, for now, maybe using chromogenic assay is a much more like the conservative way, and potentially the upside, it depends on how aging recognize the measurement?
Yeah. I think it's fair to say it's at a minimum a more conservative way of looking at things, and it really again pertains to the accelerated approval consideration. But the part of the reason that we were willing to adopt more conservatives, because we also knew what the results of the phase 1/2 study were at similar time points using both assays, and we would have tuned our plan a little bit when we switched from one stage to chromogenic for power perspectives if we thought we needed that. And so when I say the study is adequately powered, it was adequately powered based on our chromogenic results from the earlier study.
Okay. And then if we translate into, say, the 1 year and 2 year data into chromogenic assay using, like, say, roughly 60% conversion, is it fair to say, like, the first year actually showed about close to 60%, and the second year is at 26%?
Off the top of my head, well, so first of all, the numbers for year one sound about right, but the key thing I want to point out to you is none of that matters. That doesn't matter for full approval, 1 year, 2 year, and it doesn't matter for accelerated approval, which will be determined week 23 to 26. So we can look at the pattern, we can try to prognosticate what's going to happen, but we are missing a lot of variables in terms of that prognostication. And I know everybody wants to predict the future with tight degrees of accuracy, but there's a lot of unknown about the future. And the point of emphasis from a regulatory perspective is short-term factor, long-term bleeding. And by long-term, I mean bleeding. Factor isn't really of significance to the FDA at week 52, but bleeding is.
Okay. So maybe going back to the 3 year data, I know a lot of investors were like focusing on this catalyst, and then you will present the data likely at the ISTH in July. So do you still plan to release the top-line data prior to the full data presentation? And what could be the timeline for the top-line data?
Well, the plan is, we made a commitment at WFH, World Federation for Hemophilia, last May at the end of the month of May, it was May 27th, I think, something like that, where we said, and we'll give you next year's update at next year's major medical conference, which happens to be ISTH. The data were maturing through the abstract deadline, and so, the target would be a late-breaking submission to ISTH, which we haven't made because the ISTH deadline is for late breakers is, I think, June 6th. So, the process is we haven't seen the data. We need to go from a state of not having seen the data to a state of having seen the data, authoring abstracts for submissions, gaining agreement with the presenting authors, and submission. When in that time period BioMarin is going to be ready to communicate, it's not any earlier than roughly where WFH was last year. It can't be. It's not going to be much later than the first week in June because we would be sitting on super material information at that point. But somewhere we haven't alerted you to the date yet.
Okay, that's very helpful. And also, with 2-year factor A level at 26%, if we use, chromogenic assay translation, what level? I know you haven't seen the data, but what level at 3 years that you would consider as a supportive of the durability?
It's a really incredibly difficult question to answer, in part because of some missing variables, not least of which I was trying to set up earlier in the conversation, which is that right now everybody is guessing about what level of factor VIII corresponds to the spigot being turned off. And so the two things that are making the answer to that question very difficult is that we have a relatively small n for trying to calculate the evolution of the factor VIII expression level over time. And that's compounded by the fact that there's both intra and inter-patient variability. So if you think about year 1 versus year 2, I don't think we ever ran a formal statistical test of year 1 versus year 2, and you'd have to adjust for multiplicity because there's about 1,000 of them that you can run. But I don't believe there's any significance to the in a statistical level between year 1 and year 2. I could be wrong about that because I'm sure you could find a statistical test that would reveal the difference. But the point is that there's a lot of variability. And so, you were just citing some numbers, but there's a lot of plus or minus associated with those numbers. And so there's no way to set a number that would constitute something that is statistically meaningfully different from year 2 to give you an answer to that question, purely based on numbers. But there's a second concept, which is that we don't know what number corresponds with bleed control. So, for example, if I told you if 15 years from now we look back in this and we said, wow, everything settled around at, I'm making this up, by the way, at 5%, and for the next 15 years there was absolutely no bleeding and nobody was back on prophylaxis. That result, by the way, is not unpredictable because that's what Nathwani saw in his factor IX longer-term data. He's out now 6 years in his high-dose group, 8 years in his medium and low-dose group. 70% of the patients remain essentially bleed-free and off prophylaxis. And in the high-dose group, it's a higher proportion of the patients. So you can get quite a bit of good, long, durable bleed control out many years past where things settled out. And I just don't know, I'm just being honest with people when I say we just don't know where it is. So the two factors that are challenging in terms of giving you a specific and concrete answer to the famous 26 number question is there's variability around that number both in year 1 and year 2, and there will be in year 3. And secondly, we don't know what the predictive reliability of that number is. And we don't want to get ahead of ourselves by putting our nickel down on a number that turns out to be the wrong number in either direction. So I think that all that nets to keep your primary focus on, is there bleed control, is there hemostatic efficacy in year 3? That's what the FDA is going to look at, that's what payers are going to look at, that's what patients are going to look at. Factor level is of interest, I get it. I'm not saying it's not at all. You're trying to calculate trajectories, but it's going to be many years before you can interpret that trajectory.
Okay, that's very helpful. So another question, you also mentioned payer. So like, I think one question maybe payer will look at, or the investor will thinking about the factor A level will be predicting, say, if the bleed rate control very well on the third year and the factor A level drop at certain level, and then the concern will be how, what about fourth year, will we still be able to control the bleed rate with, the declining factor A level? So just wondering, how important, like your discussion, I'm pretty sure now you already talked to many payers, like what is their feedback regarding durability and what kind of data they are looking for, for the pricing discussion?
By the way, you just put your finger on the pulse of a key over-under bet we have internally, which is after we reveal the 3-year data, how long will it take for the question to be, when's the 4-year data coming out? I have 1 minute in the pool, by the way, just FYI. On the question of payers, I mean, look, payers are not that different from everybody else in the world. The longer, the better, and the shorter, the worse. And, we would be naive to try to sell you a story that longer isn't better than shorter. All that said, the way I think about this is some is good, more is better. And what we've heard from patients and we've heard, and what we've heard from payers is that the magnitude of bleed control that you've demonstrated to date is eye-popping, and that is sufficient at some price level to motivate quite a lot of payers, and at some efficacy level, which if we replicate would motivate quite a few patients. And the more that we get over time, the more motivation and the more penetration we're going to get into the patient population. We haven't revealed a forecast and we haven't revealed a price, so all we can do is just sort of comment qualitatively that it, on the one hand, it makes sense that the more you have, the better. I think we are a little resistant to the idea that if we don't win in year 3, it's lights out, because I don't think that's necessarily where the patient community or the payers are. But clearly, the longer, the better.
Mm-hmm. And then maybe going back, regarding the decline, factor A level decline from week 78 to week 104. So any additional work you have done, regarding the mechanisms, and then, what, like, that, how that will help us understand the kinetic factor A expression in the long term?
Well, this is a key question for the field, not just for BioMarin, and what would that work be? And I think we've gotten to the place where we feel like everything that can be done preclinically we've done, and for example, that included some of the experimental results we showed at R&D Day, where we verified for ourselves that the vector can in fact circularize. Because that's important, because it's been known for a couple of decades now that when you have persistent expression, you have circular findable DNA. So if for some reason the Valrox that we were making wasn't able to circularize, that would have been a big problem. And we investigated to some level the alternative explanations for the evolution in the factor VIII expression profile, and don't find any definite answers. But I think the team has gotten to the place where we've done as much as we can do, non-clinically. The next step, I think, intellectually anyway, is to raise the question of, can we assess the state of the vector in humans that have been treated with the transgene product to generate knowledge about what the key determinants of durability are, based on some measured parameters of what can be recovered from patients who've been actively treated. And so we have a protocol amendment out there to do liver biopsies in patients and assess the state of the vector. Oddly, it's going slow. I think the notion of doing biopsy in the hemophilia community has caused ethical considerations to be raised that people need to chew through. They're not accustomed to thinking about a hemophilia patient as having the degree of hemostatic efficacy that these patients do. So it's taking a little longer than maybe we'd like, and I think we're going to have to live in a state where it's going to be a while before we can investigate what the critical determinants of vector persistence in individual patients is. And I go back to, and therefore the world will be guided for some time by much more simple and practical ways to interpret the long-term effectiveness, which is, is patient bleeding or not.
Mm-hmm. Okay. And then, certainly BioMarin is leading the race of hemophilia gene therapy, and we hope next year we will have a first gene therapy, hemophilia A gene therapy come to the market. But also we do see, quite some upcoming competitions. How do you, or where do you see the key differentiation, for future commercial success? And we also see like Spark and Sangamo actually just reported some data with, I think, a first cohort, 2 patients, who looks pretty promising. And what would be, where do you see the key differentiation for the future commercial success?
Yeah, so I think when I think about that, I think about safety, efficacy, cost, and convenience. Convenience, for the most part, is going to be more similar to the difference among the different flavors. People have made a big hoo-ha over the corticosteroids, and a day here, a day there is not going to really meaningfully move, product choice. Safety perspective, I do think if you have to be hospitalized for transaminitis and corticosteroid therapy, and you're losing factor expression, that will be a consideration. So I think everybody's digested the fact that problem must be overcome. But if you had efficacy in a large, a swath of patients that you tested in a clinical trial, you could be a candidate for a follow-on to BMN 270 or Valrox. The key area of, if you will, vulnerability as we sit here today for Valrox is in the patients that we're currently excluding because they're seropositive to AAV5. Now, as you know, we've also got that experiment going on to see if we can transfer the gene into AAV5 positive patients. So in some sense, we're ahead of the competition in that regard as well. But if it turns out that we can't figure out, at least for a few years, how to treat an AAV5 person, if a Sangamo or a Spark came along with a capsid that was not cross-reactive with AAV5, they might be able to nuzzle into the market. We've also thought about other places, other spots in the hemophilia landscape that companies could land and develop some market share. And we don't feel like there's an easy way for companies to go past, for example, demonstrating factor expression in severe patients with hemophilia A without inhibitors. And what I mean by that is you notice that, for example, with the Spark program, when they switched over from 8011 to 8016, which they're calling their inhibitor program, they started in non-inhibitor patients. So everybody is behind us. The only opportunity to catch up is really in the seroprevalent, AAV5 seroprevalent population, if there is a there, which I don't know. And every other way of trying to catch up with us, we're further ahead already. So I'm pretty happy about where we are competitively. Hope that there are no, that we can keep this pace up. If we can, then we're in really good shape.
Mm-hmm. So you also mentioned that you have a phase 1/2 study hemophilia A patient with AAV5 neutralizing antibody. Just wondering, in your hand, what is your experience of factor A level and the level of pre-existing neutralizing antibody?
Yeah, we showed some data, I think it was at ASH, not this past year, but the year before, demonstrating in non-human primates that in the face of relatively low titer antibody in the range of 1 to 150, we could affect gene transfer in the majority of animals, but that gene transfer appeared to be attenuated relative to naive animals. That attenuation was maybe a cutting in half of the level of factor VIII expression off the top of my head. And not in 100% of the treated primates, non-human primates. The key question is going to be, how does that translate into humans? And for that, so there's enough reason to believe that it could happen, but we have to see to what degree and to what extent in the population, and that's the subject of the ongoing study.
Okay, that's very helpful. I think that, we also know that Unicures are using AAV5, and in their experience, actually, seems like factor 9 level, and that's their hemophilia B program, seems to have no correlation with pre-existing neutralizing antibody. Just, would like to hear your thoughts. Is that, do you think that that's AAV5 specific, or what might be the mechanism?
Well, they're doing my understanding is they've basically done and are continuing to do relatively all-comer studies. So 10 or 20% of their population that they treat, depending on where they draw their patients from, 25% of the population could be AAV5 positive over a wide range of neutralizing antibodies. And so when you think about subgroup analysis of 10% or 20% of the population, it could be difficult to discern from that data set exactly what the impact of pre-existing antibodies is. But I will say, to your point, we are encouraged that they are willing to do that, because it conveys independent scientific credibility around the notion that pre-existing immunity isn't the complete black-white stuff that people had been thinking that it was. By the way, while you're on the subject of Unicure, I should mention that they presented at ASH last year some updated data on 060, which is their predecessor transgene for factor 9. And they actually have factor durability data out now 130 weeks, well past the famous 2-year mark, and bleed control out past that 2-year mark. So there's been a lot of, there's been wonderment and shade, if you will, thrown on AAV5 and bacular expression, and I think that more and more you're going to see companies working out of baculo and demonstrating that baculo is a good expression vehicle, and you'll see other transgene products that have evidence of durability of expression, so it won't just be all based on what BioMarin is seeing.
Mm-hmm. Okay, and then maybe I just ask one more scientific question regarding the factor A level expression. I think that Sanghamon's data, although it's only 2 patients at their high dose, seems they have very rapid uptake on the factor A level expression compared to BioMarin. And then, so from scientific point of view, what might cause these differences?
I think people think that capsids, play a pretty significant role in the kinetics of expression, because different capsids are internized, uncoded, and DNA repaired at different rates. And so I think the prevailing hypothesis I've heard is, is capsid, but who knows?
Mm-hmm. Okay. So I will switch gear, maybe asking about manufacturing, and these questions are mainly for Rob. Maybe, first question is, I think you comment your current capacity can supply 4,000 patients at the highest dose. Can you walk us through the underlying assumptions, these 4,000 patients?
Sure. Yeah, sure. Thank you, Gina, for the opportunity to talk with you today. As you may be aware, in 2016, we decided to build our own manufacturing facility. This is based on our long experience in producing biologics products, going back to the approval of Aldurazyme in 2003. And so we built a facility anticipating what the demand for not only factor VIII, but other gene therapy programs that we have. And so when we built the facility, we looked at a number of things. We had data already on the baculo expression system. We certainly have learned over time in terms of how to increase the productivity of that system. And we designed the facility to run a multiple 2,000-liter bioreactors. And so our combination of the titers that we attain for the production, the scale at 2,000 liters, and then matching purification for that is how we arrived at the 4,000 patient number.
Okay. And then I think that you also said at some point wanted to improve this to 5,000 patient capacity. So how would you, which are the factors you may need to improve in order to reach higher capacity?
Yeah, so there's two components to that, and they're relatively straightforward. One is adding additional equipment in the facility, and there is existing space for that. So as you might imagine, as we build out the facility, we can incrementally add additional equipment to be able to go from that 4,000 to 5,000, and there'd be some incremental headcount add to accommodate that higher production rate. And based on our history to date and projections we have, we feel pretty confident of that 4,000 or 5,000 patient per year number.
Okay. And Rod, would you be able to give us a little bit more specific numbers regarding, say, how many equipment you have, and does each one has, what is the maximum capacity for each one, and how fast you can do, running, say, one production? How long does it take to run one lot of the production?
Yeah, so the production runs over several months time for each batch. We can do those concurrently from multiple bioreactors and purification systems, and so the decision on when to scale that out and is really predicated on the clinical data that Hank's just talked about, and the amount of time we have to be prepared for that. The plannings we have in place already are well matched to the clinical development plan and future commercialization plans. So we have the ability to scale out the facility by adding additional equipment, and those are relatively short lead time types of equipment, things we've purchased in the past already, and we're well suited to be able to meet that demand as the need arises to produce that 4,000 to 5,000 patient doses per year.
Okay, so for the equipment, can you share with us, the name of the bioreactor, and then so each one, how many would that be able to supply? The number in terms of the number of the patients.
Yeah, we don't typically talk about the vendors of equipment. There's only a couple of different vendors for some of these bioreactor systems. We've looked at many different sources of equipment, not only with the gene therapy, but they share some commonality with the biologics production we've done over the years. In terms of the number of doses per lot, we've not disclosed that either, and that's a level of detail I'm not exactly sure what the pertinence of that is, beyond knowing that we're committing to 4,000 or 5,000 patients per year coming out of the facility.
Okay. And then another question, just quickly. I think you mentioned in the past that you wanted to complete a manufacturing campaign required for accelerated filing that was guided by the end of March, and just wondering, have you already completed that process?
Yes, we have completed that. We had a very successful campaign for that, and we are now in the process of characterizing the material we made that will be part of a BLA submission. There's a variety of things that go into meeting FDA's requirement for demonstrating not only robustness of the process and reproducibility, but meeting very specific criteria. Those are generally all pre-specified in protocols with acceptance criteria, and so we finished the production phase of that, and we're now in the analytical characterization of that material. That's all on schedule, and again, consistent with the planning we had to be correlated to the clinical data coming out.
Mm-hmm. Rob, also another question is, I think that the field kind of split into two sector. One is like a mammalian system, and you are on the other side is a baculovirus system. So will you continue to focus on baculovirus system for future indications, and where do you see the pros and cons compared to the mammalian system?
Yeah, so when we first got into the gene therapy business, we did a thorough evaluation of the baculovirus insect cell production system as compared to the mammalian HEK 293 system that a number of companies are using. And when we looked at that, there was a variety of reasons that we wound up choosing the insect cell system. First, as I've alluded to, we've already scaled that up to 2,000 liters, and most companies that are working with mammalian cell lines are either in an adherent system, we're in suspension culture, or if they are in suspension culture, they're at a much lower volumetric level, either 200 or perhaps 500 liters. The other aspect of this is the insect cell lines grow to much higher cell densities and have higher titers on the order of 30, 40-fold higher titers. So the productivity is a combination of both higher titers as well as higher volumetric productivity. We also looked at a variety of other aspects related to using mammalian cell lines, and there were sort of two potentially safety production-related issues we thought about. Human cell lines have the potential for carrying oncogenes, and they also are much more susceptible to contamination with human viruses, and many times require serum to be used in the process that is a known source of viral contamination. And for those combinations of reasons, we chose to work with the baculovirus system. From an ongoing perspective, certainly we've had great success with that, both from a manufacturing perspective as well as what we've seen in the clinical results. And so that would lead us to continuing to use that sort of system as a platform technology unless there was some other reason to move into mammalian cells, and that would be more related to transgenes that are much larger than the AAV virus can encapsulate. So I would anticipate, as we develop other programs, to be able to leverage that technology in a very meaningful way, which has the other advantage of decreasing our overall development timelines as we develop a platform approach to our manufacturing and process development.
Okay, that's very helpful. I would just ask one last very technical question. Just wondering, in your practice or experience, what is the full versus empty capsid ratio for Varox?
Yeah, a lot of companies talk about their ratios. We really don't have one. And the reason I say that is we have developed a process that removes all the empty capsids. So we have undetectable level, and we have very sensitive assays, an undetectable level of empty capsids. And there's a lot of debate in the industry about what's the impact of empty capsids. I'm sure you're very familiar with some companies proposing empty capsids are a decoy to the immune system. We took a very different approach to that. We've said empty capsids don't have a transgene, so why would we have that impurity in our process? And we base this on some scientific data as well. We've developed a potency assay, which is a human hepatocyte-based assay in which we can transfer the gene into those cells and they can express factor VIII. And when we do those studies, we've conducted a study in which we hold the amount of full capsids at a constant level and spike in empty capsids, and what we find is a linear decrease in potency. And what that would imply is that the empty capsids are not really acting as decoys, but are competing with full capsids for receptors on the cell surface. And for that reason, we developed a process in which we remove all the empty capsids and don't really have to deal with what's our ratio and how consistent is that, and exposing patients to capsids that are not able to transfer the gene of interest.
Okay, that's very, very helpful. Thank you, Rod. I will also now switch gear, maybe quickly talk about vosoritide in achondroplasia. So I understand you have a very comprehensive development program. Just wondering, Hank, if you can, remind us, regarding the registration package for, the FDA. What are the feedbacks from the FDA?
Yeah, so 4 key components of the registration package for the achondroplasia program. One is a randomized placebo-controlled pivotal trial, 52 weeks in duration, fully enrolled, data readout towards the end of the year, beginning of next year. Second thing is ongoing long-term effectiveness data. And again, this is another example of durability questions, no statistical consideration in this, but just continued characterization of the long-term effectiveness of vosoritide, to which we've shown you 42 months worth of outcome data at R&D Day last year, and when we unblind, therefore, the pivotal trial will be at about 54 months, and clearly more, when we submit the file to the FDA. So we'll have 4 or 5 years worth of durability data in that population. Third thing that, in the registration enabling program is safety data from children who are younger than 5. As you know, our pivotal clinical trials in patients who are 5 years of age and older, and because it's a genetic condition and because most of the growth attenuation that occurs in these patients occurs in the first 5 years of life, the agency knows that the product is going to be used in patients earlier than age 5, and so safety data in the under 5 population, we're well underway. We've already cleared the sentinel cohort for the first, I think, in that second cohort of patients as well, maybe just the first cohort of patients so that we can start treating the second cohort of patients. So looking good for safety data. And then the last leg of the registration submission is, again, not statistical, but contemporaneous natural history data that addresses the question, given your durability data, how can we be confident that in the long term, the 1-year height gains that you've observed year by year add up to a taller height than the patient would have otherwise experienced if they had not been treated with vosoritide. So that fourth component, contemporaneous natural history. So all 4 pieces are in track, and we're pushing towards the pivotal component of it towards the end of the year in terms of unblinding the phase 3 trial.
Sounds good. And for the phase 3 trial, how much increase in annualized growth velocity at 1 year? Do you, based on your feedback with doctors and of course FDA, do you expect it will be clinically meaningful and also approvable?
Yeah. So the 1-year number is just part of the whole package because nobody's in this for just 1 year of outcome. And I think one of the elements of item number 2 and item number 4 in my list that I just gave you is what's it all add up to. And there's going to be a relationship between when you start and what's it all add up to. And so one way to think about that is we know that a certain group of patients are motivated to get, for example, limb lengthening with the promise of gaining, I think it is, another 10 centimeters of stature. But that benefit is maybe even a little higher than might what otherwise be expected for two reasons that 10 target. One is that the limb lengthening procedure is extremely painful, so if you're not going to get a lot of juice out of that, it isn't going to be worth the squeeze. And secondly, because the limb lengthening procedure only addresses skeletal length, doesn't address any other facet. So I don't think 10 is the minimum target of effectiveness. I think it's lower than that. But I don't think that we've arrived at a specific and concrete number. I will say that I think the medical community is extremely excited about 5.7 centimeters of additional stature gained in only 42 months worth of treatment, because projected out over 5 years of treatment, that averages, I think, 1.6 centimeters per year of additional height gain. Averaged out over 5 years, that's already at the limb lengthening length practically, 6 years for sure. So what does that mean? That means that I think basically everybody would start if they knew they had 4 or 5 years of remaining growth left in them. And there may be some people who are willing to start even later, but my gut feeling, based on what we've talked about with clinicians and whatnot, that 5.7 centimeters in 42 months is viewed as very exciting to the community, and if the expectation holds that it continues to add up, that we'll be at an unequivocally meaningful degree of height gain, relatively shortly.
Mm-hmm. Okay, that's very helpful. Operator, maybe we'll open up for any questions.
Certainly. As a reminder, if you'd like to ask an audio question, you may do so by pressing star followed by the number 1 on your telephone keypad. Again, that's star 1 to ask an audio question. We'll pause for just a moment to compile a Q&A roster. Once again, ladies and gentlemen, that's star 1 to ask an audio question.
Okay, operator, while you're waiting, maybe I will ask one question here. So that's regarding PKU franchise for Palynziq launch. How has been the safety profile in the real world?
Yeah, so far so good, but recognize it's a relatively small handful of patients. We got approved in the U.S. at the end of May, didn't have our first shipments until the beginning of July, and so as of December, which was the last, sort of the cutoff point roughly for our most recent public reporting of the data, relatively few patients had been on therapy for as long as 6 months. When we did our quarterly call, we had some awareness of what was going on into Q1, which is really when the very first wave of patients is coming through the system. So far, rates of side effects have been very consistent with what was observed in the clinical trial, if not lower. Compliance with the REMS program has been outstanding. Patient compliance has been outstanding. So all systems are green and go, with the acknowledgement that we're still relatively early in the wave of induction titration and maintenance. Now, hopefully, this watchfulness on this particular parameter is just a few years while we convert essentially as many of the in-clinic patients to Palynziq as we can, and then turn our attention to the slower process, which is going to be absorbing the out-of-clinic patients into the Palynziq practice area. So hopefully this induction titration hypersensitivity reaction rate issue is going to be over and done with really in the next year or so.
Okay, great. Operator, do we have any questions?
And at this time, I'm showing that we have no questions of the phone lines.
Thank you. So I would just complete maybe with one big picture question. So Hank and Rob, BioMarin has a fairly meaningful base business enzyme or substrate replacement therapy for rare disease, and you also have emerging gene therapy programs for hemophilia A and the PKU. How do you see the potential threat or the future dynamic that will play out to your core or base business, and what would be your strategy to prioritize your own gene therapy programs?
Maybe I'll start and Robert can chip in. I mean, big companies, bigger companies than we can be defined by therapeutic area, platform. We're a therapeutic antibody, we're a neurology company, we're a cancer company, and BioMarin is still in its late adolescence in terms of defining, where its franchise areas are. So I would say that for the near term, while we have provocative and interesting data in gene therapy, we're not really ready to turn the switch and say everything ought to be gene therapy. And as such, pipeline assets that come up for consideration can come from a world where the best tool to use is a gene therapy, and that's what we did with PKU 307. Took us a little while to figure out how to engineer all the components of it, but there's a situation where we went from enzyme to gene in one therapeutic area. But conversely, we also see opportunities outside the gene therapy space that we're interested in. And it wouldn't surprise me if the next IND after PKU gene therapy is not a gene therapy. But I think that another way of looking at this is in bigger blocks of time. Before Valrox, we didn't have any gene therapy. After Valrox, it's looking like something like every other product for the Valrox development phase is going to be a gene therapy and not a gene therapy, a gene therapy and not a gene therapy. And let's see how Valrox and PKU 307 do. They could define us as a gene therapy company, or alternatively, some of the other pipeline assets we have could identify us as based on therapeutic area. But time will tell. We don't have a corporate mandate that yet says we're going to be one thing or another thing other than a sustainably profit-growing company. That's the main mission we have right now. Rob, is there anything you'd want to add?
Yeah, I just think, the approach we've taken with our gene therapy programs is to go into patient populations where there are larger number of patients in the ultra-rare diseases. And so, our notion of should we develop our own programs for existing enzyme replacement therapies, I think is challenging from a number of technical perspectives. There's so many different cell types you need to get those enzymes into, in the brain, in the eye, throughout the other organs, and so a gene therapy approach could be significantly challenging. We've certainly had discussions and as we think about what's next for gene therapy for us, and I think in general, as you've seen with hemophilia and with PKU, we're more focused on slightly larger patient populations than the ultra-rare diseases.
Okay, that's very helpful. Thank you, Hank, and thank you, Rob, for joining us today. Also, I wanted to thank everyone who dialed in. This has been very helpful, and I will look forward to talking with you in the future. And then this concludes today's call.
Thank you, Gina. Thank you very much.
All right, thank
you.
Bye.
Bye.
Ladies and gentlemen, this does conclude today's conference call. We thank you for joining. You may now disconnect.
Hope this gets accurate. Hope this gets accurate.